Hello everyone, and welcome to the next webinar in the Thorlabs How to Build a Microscope series. My name is Eric Leeser and I'll be moderating today's webinar. In this webinar, Jeff Brooker and Brian Mell will discuss a few methods for extending the function of a multi-photon microscope. The topics today will be fluorescence lifetime, 3-photon, and Bessel beam imaging. Jeff Brooker is the Life Science CTO for Thorlabs, joining in 2008 and starting the Imaging Systems Group located in Sterling, Virginia. The group is focused on developing and manufacturing tools for microscope imaging. Currently, he's responsible for running the Advanced Imaging Research Group, which focuses on collaborations with microscopy thought leaders in academia and the development of next-generation imaging systems for the Thorlabs product line. Dr. Brian Mell is a senior systems engineer in the Advanced Imaging Research Group, joining Thorlabs after working at Howard Hughes Medical Institute, Genili campus in the Transcription Imaging Consortium. As an instrument designer and builder, he has developed imaging technology in fields from physical chemistry, cell biology, and neuroscience. He continues to lead collaborations on the development of advanced techniques in his current role. Please feel free to ask us questions throughout the presentation using the question and answer tool. The presenters will address your questions at the end of the presentation. I'll now hand the talk off to Jeff Brooker.
Thank you, Eric, and good morning, good afternoon, good evening. Thank you for joining everyone. Today we're going to talk about three different areas, as Eric said. First, we're going to discuss 2-photon FLIM imaging and a unique implementation that Thorlabs has developed. Then we're going to move on to discuss 3-photon imaging for deep tissue structural imaging. And then we're going to talk about extending the depth of field of multi-photon imaging using a Bessel beam. But first, we're going to start with 2-photon FLIM. And I'll start with a little background on FLIM and highlight the differences between standard 2-photon imaging and 2-photon lifetime imaging. So the top image here is what we typically expect to see from a traditional multi-photon image of a piece of tissue. It's a grayscale intensity-based image that gives structural information about the sample that it's being observed. And it can also be used to measure biological changes over time. The second image below is a pseudo-colored representation of the same tissue in the same fluorochrome. And as you can see, there's a significant difference in the lifetime of the fluorescence, which shows on a point-by-point basis and shows different metabolic states within the sample. The FLIM image allows us to distinguish different environmental conditions on a pulse-by-pulse basis at any given point in time. So now let's go to the standard energy diagram for multi-photon fluorescence. As we usually see, the red line represents the emitted photon from the laser. In 2-photon excitation, there needs to be 2 photons from the laser very close in time and space to elevate the energy state of a single fluorescence molecule. And then there is a period of fluorescence decay for the molecule to return to its ground state. In FLIM, we're monitoring the time it takes for the molecule to return to its ground state after being excited by the laser pulse. This is rate dependent upon the other non-radiated processes that are going on at the same time. A really good way to think about this is to think of fluorescence as a bucket of water. We monitor the visible emission, which represents one of the ways in which the molecules are relaxing. The other non-radiative paths that we can't see have a significant effect on the speed with which the fluorescence decay occurs. The other paths can be thought of as various size holes in the bucket. The larger the non-radiative path is, the quicker the bucket empties. Fluorescence decay of a pure molecule is a known constant, so when we monitor the fluorescence in the sample, it's directly proportional to the rate with which the total decay occurs. Different decays represent different metabolic or environmental conditions. This gives us a point-by-point functional view of what's really happening in the sample. So what can we measure with FLIM? Lifetime depends on all kinds of things inside of a living tissue or a cell. We can look at oxygen concentration, we can tell what the pH is, or we can even look at changes in protein conformation over time. All of these changes can be used as a biosensor to monitor conditions or processes in living cells and tissues. So this movie right here is a great example of how we can use FLIM to see metabolic transients occurring in live neurons. There's a brief increase in the RCamp signal, which shows a calcium spike after the firing of a neuron, followed almost a minute later by a change in NAD/NADH concentration. This redox reaction is significant in cellular respiration and would otherwise be missed if one was only just looking at the change in calcium. For reference, the images are pseudocolored according to the sensor lifetimes, which is shown on the scale bars on the sides. Furthermore, we can calibrate this to show the ratio between NAD and NADH at any given point in time. So, how's lifetime measured? Here, we have a representation of a laser pulse. And when the pulse occurs, it excites the fluorescent molecule, and then that molecule emits a photon shown by the green dot. We record the time between the laser pulse and the emitted photons, and then use that data to build up a histogram, which statistically represents the decay curve of a given molecule for a specific time range. We typically use a laser with a repetition rate of 80 megahertz, which gives us a period of 12.5 nanoseconds between pulses to observe the fluorescence decay. So classically, a time-to-digital converter is used to measure the interval between the laser pulse and the arrival of the photon. A TDC can be thought of as a stopwatch. The laser pulse starts the stopwatch, and then the arrival of the photon at the photodetector stops the stopwatch. During the readout after the event, the time difference is communicated to the computer. The temporal resolution of these devices is very high and can reach 10 picoseconds, but there's some inherent problems for this application. The two major issues with this method are dead time and pulse pileup. The first issue, which is dead time, comes from the fact that the TDC circuit needs to rearm itself between measurements. This can take between 80 to 125 nanoseconds. While this really doesn't seem to be a long time, the system is totally blind for 6 to 8 laser pulses while this is going on. And this has a significant impact on the speed with which we can collect the data. The other major issue is called pulse pileup. In the cartoon here, we can see that some laser cycles have 1 photon, some laser cycles have no photons, and some laser cycles have 2 or more photons. It's dependent on a lot of things like the intensity of the laser, the fluorochrome concentration, and even the sensitivity of the detector. When there's more than 1 photon per laser pulse, the TDC can't discriminate between the first and the second photon, and therefore only registers 1 event. So to solve some of these problems, we've developed an all-digital FLIM solution. First, we take and we convert the laser pulses into a clock signal that we phase lock loop to a 1.25 gigahertz timing pulse. This triggers a fast analog to digital converter on our FPGA board in the computer. By continuously sampling at this high rate, we take 4 to 5 8-bit samples for every photon emitted, and there's no dead time between the measurements. The additional advantage is that we can sum the total photons to create an intensity image comparable to a traditional 2P image, 2P system. So additionally, because we're counting every photon that's emitted between laser cycles, we can tell if there's more than 1 photon has arrived in a given period. This allows us to eliminate the bias that's usually generated by pulse pileup if the intensity is too high. We simply eliminate any pulses in our calculations where more than 1 photon has arrived. Additionally, we do a curve fit in the FPGA for every pulse that is detected, And this increases the resolution of our system from 80 picoseconds, which would be the native resolution of a 1.25 gigahertz sampling rate, to 50 picoseconds, which would be equivalent to sampling at 20 megahertz. So now to output the data, we can take the total photons counted for a given pixel and generate an intensity image like this, which is what we would get from a traditional 2P system. But since our data also includes the mean arrival times for every single photon captured, we can show the pixels as a heat map. The color in the image correlates to the lifetime recorded for each pixel in the field. And as you can see in this image, the structure looks the same, but there's a significant amount of difference in the lifetime of the fluorochromes across the image. This is key for unlocking additional functional information about the sample. So how do we do this? Well, let's look at the component of a typical 2P system. There's, of course, a femtosecond laser, scanners, and a microscope, which has a non-descanned gas detector coupled to a digitizer in a computer. For our FLIM system, however, we changed the detectors to a hybrid detector that has a better transit time and specific pulse shape on the output. And then we changed to a much faster digitizer in the computer. We also add a laser sync pulse that gives us an exact reference as to the laser pulse, so we can calculate the precise arrival time of each photon. So to summarize, 2-photon FLIM gives you functional information in samples that's not otherwise available from other methods. Different relaxation pathways can be measured with great precision. And our all-digital implementation has the additional advantage of high acquisition speeds as well as no dead time between measurement cycles. We can also provide a traditional intensity image, which is not typically available with TCSPC methods. Finally, our setup is simple and easy and doesn't require the tuning of fixed delays that come with the inherent problems related to cable lengths and electronic transit times. So for both thin and thick samples now, we've created an ideal system for analyzing both structural and functional information from your samples. So now my colleague Brian Mail will continue the talk and discuss 3-photon imaging as well as an exciting implementation of Bessel beam imaging.
Hello everyone, I'm Brian. I'm a systems engineer in the Virginia Imaging Research Group here in Sterling, Virginia. And today I'm going to introduce 2 additional modalities, as Jeff mentioned, whose foundations are really built upon multi-photon laser scanning systems. So the first modality that I will introduce today is 3-photon imaging, or 3P, which is really well suited for deeper imaging in scattering tissue. So 3-photon imaging deeper in tissue. So if we look here to the left here, this is actually a 2P image of some mouse cortex. And what I want to point out is if you look at the top here, you can see that the contrast is very, very high. But as you go deeper into the sample, the contrast sort of deteriorates and it becomes very, very low. You can quantify this by giving it sort of a number called signal to background ratio, where at some point you can go deep enough where the signal to background ratio approaches 1. And this is when the signal is just as high as the background, and so you really just don't get any information out. And 2-photon, this can occur somewhere between 450 and 850 microns in the mouse cortex when using 920 nanometer light. So if you want to have better resolution deeper, you need to maximize your signal, so effectively get excitation photons to the focus, and you want to minimize the background, so removing any sort of additional background fluorescence. 3-photon microscopy achieves this by using longer wavelengths and higher order nonlinear excitation. By higher order nonlinear excitation, I mean the fluorophore is absorbing 3 photons simultaneously as opposed to 2 photons simultaneously. So shown here on the left is a 3-photon image of a mouse cortex in vivo that was imaged through a cranial window, courtesy of Cornell University. And the thing to sort of point out is even at 1.2 millimeters, you still have a good signal to background ratio. This was taken on a Thorlabs Bergamo 2 Galvo system. And then the main difference between using a 2P laser imaging system versus a 3P imaging system is really the laser. And so this is a 3P image, and so they used a laser that was at 1300 nanometers, so longer wavelength than typical 2P, and a low duty cycle laser, so shorter pulse lengths and shorter repetition rates. So for anyone who's interested in more information that's going to be described here today, I highly recommend to check out this review article from leaders in the field called 3-Photon Neural Imaging and Deep Mouse Brain that was published in Optica just this past year. And I know that this is being recorded, so you don't need to write this down. You can always just go back to the internet and find these references. Nevertheless, so the thing we're going to kind of focus on is sort of the laser system and how these longer wavelengths and higher order nonlinear excitation give you this better signal to background ratio that allows you to image deeper in tissue. So longer excitation wavelengths. So longer excitation wavelengths have longer attenuation lengths. This results in less tissue scattering. So let's define what an attenuation length is. So if we look at this pictorial on the left here, the attenuation length is just the depth at which your initial power has been reduced to 37%. So another way of saying this is if you had 100 photons at the surface, where's the depth that only 37 of them remained? And so things that can attenuate power can be scattering. So a photon can scatter. So plotted here on the right is attenuation length as a function of wavelength. And if we look at sort of a typical 2-photon wavelength, 920, we can see that attenuation length is around 175 microns. As we go farther to the IR, 1300 nanometers, we can see that attenuation length increases all the way up to 350. If we go even farther to 1700, that attenuation length can be pushed all the way up to 650. But this isn't the only mechanism that can attenuate photons. You can also get absorption. And so, unfortunately, longer wavelengths also have more water absorption. And in tissue, which is a lot of water, can result in more tissue heating. And this ultimately limits the power allowed for imaging. Nevertheless, you can combine these two mechanisms to get an effective attenuation. And this sort of opens up two spectral windows to do 3P imaging. The first, sort of around 1250 to 1400, overlaps nicely with GFP and YFP fluorophores, where the second window, 1650 to 1850, overlap nicely with red fluorescent proteins. I should say that this chart was just modified from this Nature Photonics paper that was published in 2013. So how do you improve the signal to background ratio? I think the best example showing this is this figure from this paper, Nature Methods paper in 2017, where if you look at this top panel, this is a 3-photon image taken at 1300 nanometers. And then if you look at the bottom panel, it's a 2-photon image of the same sample at 920 nanometers. And both these were taken at a depth of 780 microns. Just qualitatively, to me, it seems clear that there's basically no background in the 3-photon image, and while there's sort of some background in the 2-photon image, and it looks staticky. You can quantify this by taking a line profile shown in yellow and plot that on the right here. And so if you normalize for the signal, and then you look at sort of the blue curve versus the pink curve, you can see that the blue curve, the background of the 3-photon, is orders of magnitude less than the 2-photon. So 3-photon excitation dramatically reduces the out of focus background, improving this signal to background ratio by orders of magnitude. 3-photon excitation provides significant improvement in the overall excitation localization. Another way of saying this is that 3-photon has a better optical section capabilities compared to 2-photon. So 3-photon excitation can be significantly increased by using a low duty cycle, high pulse energy excitation source. So if we're going to do multi-photon excitation, this really requires more photons to be absorbed simultaneously. And this ultimately makes the likely that this event to occur more uncommon. And this is quantitatively given by this excitation cross-section. If we look here to the right, if anyone's sort of shine light into a fluorescein sample, you sort of can get one photon sort of excitation, and you can see emission coming throughout the whole sample. And this is why when you do confocal microscopy, you need a pinhole to get any sort of optimal sectioning. Using 2-photon, a nonlinear process, you can sort of remove this the necessity for having a pinhole, because the likely for this to occur only happens when a lot of photons arrive in a short period of time. When we jump to 3P, this is even a more rare event, or a rare event to occur, and this excitation cross-section is lower than even 2P. So this really necessitates having high peak power to ensure multi-photon excitation. You need a lot of photons to arrive in a short period of time. So if we look at sort of what a traditional 2-photon laser looks like, it generally sort of is an 80 megahertz rep rate, 100 femtosecond pulses. But if we need more photons to arrive in a short period of time, there's two ways that we can get there. The first thing is you can shorten the time in which the photons arrive, so 100 femtoseconds to 50 femtoseconds, and you can have more photons arrive per pulse. But in order to have the same average power, you need to be able to change this rep rate. So a 3-photon laser will have higher pulse energy for the same average power compared to a 2-photon laser. I suggest anyone interested in more depth, detailed understanding to review a previous webinar called Selecting the Right Ultrafast Laser in Optics, which I thought was very good at sort of describing this. Okay. So now that we're going to use these low duty cycle lasers, there kind of needs to be some consideration on how you collect your data. For an 80 megahertz rep laser, you have a laser pulse coming every 12.5 nanoseconds, shown here in red. But your sample is decaying in a handful of nanoseconds. As for 3P, or so for a 1 megahertz rep rate laser, the laser pulse is coming every 1,000 nanoseconds or 1 microsecond. So if your fluorescence is still only decaying within a couple nanoseconds and you're sampling over this whole window, you're sampling a lot of time when nothing is occurring. So generally, it's good practice to come up with a digitizer scheme for these low rep rate lasers, where you're selectively only sampling when the fluorophore is decaying and not over the entire pulse, between pulses. Other challenges and opportunities for 3P. So I hope I've gotten across that 3P really requires these high power, short pulse width, low repetition rate lasers that are tunable from 1300 to 1700 nanometers. So this sort of also necessitates that your optics, your scan lens, tube lens, and objectives are also corrected for these wavelengths and can efficiently transmit these wavelengths. Since you need a lot of photons to arrive in a short period of time, and using these short pulses, dispersion compensation also becomes more critical when compared to 2-photon. This also is seen for power control as well, where the signal has a cubic dependence on pulse power. Current implementations are using basically galvo-galvo scanners, and so you're sort of limited to sort of approximately 8 frames per second. This is quite a bit slower than galvo resonance scanners that are used for traditional 2P imaging, but nevertheless, I'm sure people will figure out how to go faster with 3P. So that sort of finishes up what I wanted to talk about introducing 3P. Next, I'm going to move on to extended depth of field, Bessel scanning, which is really a way to get faster volumetric imaging than a traditional Z-stack. So shown here on the right is a traditional Z-stack taken with a Gaussian beam. And what this video is showing is 50 slices that were taken at 1 micron step sizes. And this is courtesy of this advanced imaging microscopy workshop in 2019. And this data was collected by group members from Najee's lab at Berkeley and Thorlabs. You can take a Z projection of this data and color code the depths at which these neurons exist. So if you wanted to get functional information from this slice, there's time delay between what stack you're imaging. And so what a Bessel imaging is trying to do is eliminate that time. And so here's an example of a Bessel scan. So now we've taken one slice that's 50 microns thick. So capturing a volume information in a single scan. And here down below are some good references for anyone looking for more information on how to do this than what I'm going to present today. So now we know that a Bessel scan captures this volume information in a single scan. How does it do that? Oh, sorry. The main point is that a Gaussian stack, you gain depth information, but you lose speed, whereas the Bessel scan, you lose depth information, but you gain speed. So how do we do this? So if you look at sort of a Gaussian stack, you're really scanning with a point, and this is just sort of a representative point spread function of this. To do a Bessel scan, what you're doing is really sort of elongating this focus in Z. So you're more scanning with like a pencil. So if you want to scan with this elongated focus, how do you do that? So what you need to do is scan with what is kind of referred to as a Bessel beam. So having this annular ring that you project to the back aperture of your objective to give you this elongated focus. A true Bessel beam is infinitely thin, but doesn't really exist in practice. And so what you find is that these Bessel beams don't have all the energy inside the central lobe, and they actually have side lobes. And this kind of leads to sort of higher background and some blur in your images. So to reduce this, you can kind of play with the parameter space. And so the two parameters you can play with, the first being the thickness of this Bessel beam. So you can increase the thickness of this Bessel beam to have more Gaussian-like character in the beam and have at the expense of having less axial extent. The second parameter that you can kind of play with is numerical aperture, where you sort of lose lateral resolution, but you sort of minimize these side lobes. How do you optimize this excitation spot? So the first is you want to figure out what kind of NA you want to use, lateral resolution, and this will be dictated by this outer diameter of the annular ring, where larger NAs will have better resolution, but more energy will be distributed through these side lobes. The axial extent, or the elongated focus, will be dictated by this inner diameter, this DI, where longer axial extent will scan larger volumes, but also at the expense of having more energy residing in the side lobes. So the rule of thumb is you want to use only enough NA to resolve your sample and use only enough axial extent to image your volume. So a nice kind of sweet spot that a lot of the applications have been using is this sort of 0.4 to 0.6 NA and an axial extent of 30 to 70 microns. So now that we know we need this annular ring, how do we generate our Gaussian beam that's coming from our laser to this Bessel beam? And the way that Thorlabs is currently implementing it is with a spatial light modulator. And so the spatial light modulator is you can apply any sort of phase pattern to make any sort of arbitrary Bessel beam. And this generally looks like a series of concentric circles that are 0 and pi out of phase. And so what happens is you send your laser beam to the SLM, which this phase pattern, which gets reflected, that diffracts. You use this lens then to image at the focus to generate this Bessel beam. Unfortunately, the SLM isn't 100% diffraction efficient, and so you'll have parts of your beam that don't get diffracted, or you'll have some energy sort of in this higher order modes, and so you need to use sort of a mask to get rid of those use this mask as a spatial filter to get rid of these unwanted sources. I found really helpful this paper here has this great code that sort of plays with all this parameter space of how you would design what you would want to put on your SLM, and really plays with all that parameter space to help you further understand how to generate the Bessel beam that you need. So how do you add this to a traditional 2P setup? So shown here in yellow is the traditional sort of Gaussian beam path, where you have a couple of mirrors, and then which goes to your microscope, which has the scanners, scan lens, tube lens, and objective. If you want to add a Bessel beam path to this, you just need a way to divert the light. And so you can either use removable mirrors or a rotating halfway plate with a polarizing beam splitter. So then you would send your light to this SLM that has your phase pattern that gets diffracted and imaged with the lens to this imaging plane here, where now you have your annular mask that spatially filters or blocks all this non-diffracted light. You then can use a pair of lenses to conjugate to these scanners. And so in doing this, having this additional module kind of gives you the best of both worlds, where you can do get the structural information with your Gaussian beam, get all the depth information you need, and then do the functional imaging with a single scan, capturing a single scan, single volume with a single scan. So considerations. So it's really important to optimize your extended depth of field for your specific sample and setup, really considering the trade-offs of lateral resolution, axial length, and power. This technique is really designed for low density samples where overlapping fluorophores will be masked in the image. Overall, this is a very powerful technique to speed up volume acquisition with minimal additional setup to a typical 2P imaging system. So in summary, Jeff talked about at first fluorescent lifetime imaging microscopy, and I followed up talking about 3P and Bessel beam, all of which are just extensions of this framework of a laser scanning multi-photon system. I hope this was informative, and I'd like to thank you guys all for joining, and we will be happy to answer any questions.
Thank you very much for those informative presentations, Jeff and Brian. We've had a few questions come in so far. I'd like to invite anybody else out there who has questions to go ahead and ask those in the question and answer section. First question came in back on the DFLIM, that video showing the changes over time, saying that there's the intensity changes you can see over milliseconds and the lifetime change over nanoseconds. Do the different X camp fluorophores, the G camp and R camp, do they have different FLIM lifetimes as well as the intensity changes?
Yeah, I'll take that question. My understanding is there are different FLIM lifetimes for the different X camps, and they can be mixed together, and it allows you to do some separation in the image.
All right, thank you. There are a couple of people actually asked this a similar question here. What about doing FLIM in combination with 3-photon?
Yeah, it's certainly possible because we're really only looking at the decay of the fluorochrome. So certainly we should be able to look at 3-photon also.
Probably this one goes over to Brian. Are the mechanisms of 2P background production different from 3-photon? I.e., the different background molecules in autofluorescence behave differently in 2-photon or 3-photon?
That's a good question. So I actually don't know the answer to that. Yeah.
All right. Then back to FLIM, what is the maximum amount of photons that can be binned per pulse for FLIM measurements with this technique, with the digital FLIM technique? And is there a limitation on photon flux for bright samples?
So definitely there is a limit of photon flux because you really want to look at 1 photon per cycle. But in terms of binning, Eric, any?
Yeah, so I'm moderating this, but I've also worked on this product. For doing the curve fitting to get the lifetime, about the maximum number we can do before the pileup starts to mean we can't curve fit is about 30 million photons per second. But because we're doing it digitally, we can count more than 1 photon for the intensity image per laser cycle. So if you have a bright section, you might not get quite as many lifetime measurements, but you can still see the intensity that happens there. And then another one for Brian. How does the laser not burn the SLM when you're doing multi-photon on or 2-photon on a SLM for the Bessel?
Can you repeat the question?
How does the laser not burn the SLM? Isn't there a lot of power in the 2-photon laser? And what's the power handling of an SLM?
What is the power handling of the SLM? I do not know what the power handling of the SLM, although it's not been a problem for us at this point.
And we typically spread the
yeah,
SLM itself is large, and we typically spread the beam out over the entire surface of the SLM. And so the actual intensity at any given small, point or pixel on the SLM is actually not that high.
Okay, thanks. In that same vein, what technology is used for the SLM?
The SLM is a liquid crystal.
Okay. And is it possible to do FLIM with a 1-photon laser?
It is, however, typically it's done with a slower laser in the picosecond range, and then the decay is measured afterwards. But of course, with a picosecond laser, the 2-photon effect doesn't occur. This is more of a CW excitation and is just pulsed.
Okay. That looks like all the questions that have come in. So I'd like to thank you guys again. Thanks, Jeff Brooker and Dr. Brian Mell for the presentation. If you have further questions that come up, you can send them over to imagingtechsupport@thorlabs.com, and we'd be happy to follow up. Thank you very much for your attendance, and we hope to see you again.